From Microbe TV, this is TWiP, This Week in Parasitism, episode 132, recorded on April 25th, 2017. Hi everybody, I'm Vincent Racaniello, and joining me right here in a wet, cold, and rainy New York City, Dickson Despommier.
Hello, Vincent.
Miserable day.
Not a great day, that's for sure.
Well, we need rain, so we
The hydrological cycle is alive, let's put it that way. It's been very active lately.
And it's totally cloudy.
It is.
Outside, my windows are covered with rain.
And it was very windy this morning also.
Also joining us from a remote, undisclosed location, Daniel Griffin.
Hello, everybody, from my remote, undisclosed location.
Is it also raining in your remote location?
I do not see any rain here.
Nice.
Really?
There's no windows in my remote, undisclosed formation.
He must be so far away that he's in a different
away
weather system, right?
That's right.
All right. Well, Daniel's not with us today. He's elsewhere, as you've heard, and but we can see him.
Yes.
He can see us, so...
He's responding from cyberspace.
All is good.
We can all hear everybody.
That's actually the most important thing that we hear, right?
Yes. We actually do see him, too, but we don't know where he is that we're looking.
Looks like he's in a room, though.
He is in a room. Locked in a room, and I can see him.
I want to tell you, before we start talking about our case, I want to tell you about the ASM, the American Society for Microbiology Grant Writing Online Course. You can participate in this course to receive an overview of the funding landscape. It's more like a canyon, isn't it, Dickson?
Just about.
Learn what makes grants successful and receive personalized feedback on your grant. It's a 7-part series taking place in the fall of 2017. There is a registration deadline of July 15th. You can go to bit.ly/ASMGWOC17. It's all one word. It's not really a word, just a collection of letters and numbers, but it's ASMGWOC17. And you can find out more, how you can register and so forth. All right, Daniel, let's resume discussing our case from TWiP 131.
Yes, let me remind everybody and then acquaint everyone clicking in for the first time. This was a case of a 39-year-old man from Thailand who reported 7 months of coughing up bright red blood. He says he's been doing this several times. There's mucus, but there's actually blood in the mucus. But otherwise, he feels well. He hasn't lost any weight. He feels reasonably healthy. He is a fisherman, and we found out that he's a fisherman of freshwater area. And he, like many other people in Thailand, is a fan of salted crab sumtum. He has no past medical or surgical problems, doesn't take any medicines. But when this continued, he came to the big city to be seen. When we see this gentleman, his exam is relatively unremarkable, except when we go ahead and do a chest x-ray, he has an area of increased opacification on the chest x-ray. And we just sort of got a few little bits of extra information at the end that he was not a smoker and did not have any toxic habits.
It was a short one.
It was a short one, but I think Dickson felt like this was enough.
For me it was, but...
Yeah, Dickson's the expert.
We will see.
He's just the world's greatest parasitologist.
That doesn't mean that at all. Stop it.
Are you denying it?
Of course I'm denying it.
Who is the world's greatest parasitologist?
It depends on what parasite you're talking about, because it's such a diverse field. That,
it can't be any
for instance, you're the world's greatest polio virus expert.
No, I'm not.
But listen to how many viruses there are, so you wouldn't say you're the world's greatest virologist, would you?
No, but I would say David Baltimore is.
I see. Because there are 7 ways for the gene to reproduce. That's true.
7 ways.
That's true.
All right, we have a bunch of case guesses.
We do.
The first is from Daniel, who is obviously not Daniel Griffin, but has the same name. Dear Tripartite, I enjoy your podcasts, especially TWiP. I am very familiar with Dr. Despommier's background, that of a medical laboratorian. I have been teaching diagnostic microbiology, parasitology, mycology, virology to medical laboratory science, MLS, and medical laboratory technician, MLT students for over 25 years. He and Dr. Griffin often give a shout-out to all the hardworking people behind the scene who help clinicians. Next week, April 23rd through the 29th, which is right now, we celebrate National Medical Laboratory Professionals Week.
All right.
The general public hardly knows we exist, but we supply critical information that a physician can use to make life-saving medical decisions. And right now, there's a nationwide shortage of qualified MLS and MLT. Your listeners can find lots of great information at the American Society for Clinical Laboratory Science website. We'll put a link to that in the show notes. Thanks again for the great podcast. I'm still waiting for This Week in Fungi. By the way, I'll guess Paragonomus westermanni for this week's case. I introduced this one in my medical parasitology course, so if I get it wrong, I will not hear the end of it from my students.
Right.
Daniel is a associate professor at Ferris State University in Big Rapids, Michigan.
Nice.
Now listen, Daniel, it just so happens that on the last episode of TWiP this week in microbiology, we celebrated National Medical Laboratory Professionals Week by having a clinical microbiologist on the show. Check it out. It's TWiP 150, microbe.tv/TWiP. And our guest was Robin Patel from the Mayo Clinic, and she talked all about what lab professionals do in her lab and so forth. So it was a lot of fun.
Here.
Check that out. Dickson.
Convergent evolution works.
What does that mean?
That means that things come together and,
Yeah, I know, but what does it have to do with anything we're saying?
No, you just told the audience that just last week you mentioned the same thing. Okay, David writes, Dear TWiP panelists, that's a good play on words. This case with hemoptysis appears to be straightforward. It's hard to imagine that the subject of this case was not worried right away when he coughed up blood. He must have abstained from visiting a doctor right away out of anxiety for the results until eventually the duration of the symptoms and his family members' constant nagging to get professional help won him over. The culprit is seemingly a lung fluke of the Paragonomus species. Now that's the way to put it, because without seeing that particular parasite and looking at it under a microscope for all of its nuance, there are lots of species of Paragonomus out there, right? So Parasitic Diseases 6th edition describes Paragonomus westermanni, but I am quite considering whether it could have been P. siamensis given the geographic location. The symptoms match, and the eating of crab has likely played a role in becoming infected. Treatment would be praziquantel or albendazole. I wonder if his postponing his visit had seriously worsened the outcomes, but it cannot have been helped. It is a real hot day in Nicaragua right now, with temperatures getting near 100 degrees Fahrenheit. Lately, I've tried to feel what temperatures in Fahrenheit are like, as Vincent recommended in TWiP, as I spent my whole life feeling in Celsius instead. Thank you so much for your show. It is so much more than just a podcast.
I love that tagline. So much more than just a podcast.
Here.
That we have to somehow brand that all over the place. What a great brand that is.
Yeah.
Daniel.
I guess you could. Twix, the Twix universe, so much more than a set of podcasts.
That's right.
There you go.
No, it's actually funny, the comment about Fahrenheit and Celsius. I had a patient today who actually had quite high fevers, but now they were, slightly above 100 degrees Fahrenheit. And so the medical team that I was consulting for asked me, are you okay with this? And I said, you realize that in the Celsius world, that's still 38. Yeah. That's still 38.9. So it's not a fever. It's all okay.
Yeah.
We have, we work in round numbers. It was 7 days a week of things. So, all right. Joseph writes, Dear TWiP team, my guess for this case would be Paragonomiasis, which he most likely obtained from his diet of somtum. Diagnosis could be made from the findings of eggs within the sputum. While you could do a BAL in this patient, I think that a sputum would probably be a better choice. Praziquantel for 3 days would be curative. I think I'll just BAL for our listeners is a bronchoalveolar lavage. And that's where you actually take a bronchoscope. So it's a small scope, say about as thick around as your well, my pinky finger. Other people might have fatter pinky fingers. But you pass it down into the lungs. You actually wedge it into one of the bronchi. You put in fluid, and then you actually, saline with a syringe, you suck it back out. And that's a way, I'll say, quite often in the developed world that we get samples. So a BAL, bronchoalveolar lavage. Anyway, to continue, in this case, once I had the results from the sputum, I probably would not test for TB, but would it be a good idea to get a PPD or quantiferon gold along with the sputum initially? Mm-hmm. Sincerely, Josef Davidoff, Hofstra Northwell School of Medicine, class of 2018.
Nice.
Nita writes, greetings. Hello, TWiP-tastic peeps. This is Nita, the Hopeful Neurology MS4. I did a happy little dance when I listened to the last podcast and got brown recluse correct. I didn't get the correct species, though. For this case, with our Thai man who is obsessed with crab sumtum, my primary differential is the lung fluke paragonomyosis. P. westermani is common in Asia Pacific and can infect those who consume undercooked crab or crayfish. The larva penetrates the intestines and matures, eventually returning back into the abdominal cavity to penetrate into the diaphragm. Initial signs can include diarrhea, abdominal pain, chest pain, and fatigue. Eventually, when the fluke enters the pleural space, dry cough that can become blood-tinged can occur. The signs can mimic TB, so ruling this out for our patient would be important. Paragonomyosis can enter the CNS and eventually cause meningitis. Diagnosis is made through sputum or feces. Treatment is triclabendazole, I believe. Other crab-related parasites include Angiostrongylus that cause bacterial eosinophilic meningitis. This resolves spontaneously usually, and I don't think dry cough is usually a classic sign. Of course, the sexy guess for this case would be Saculina, the castrating barnacle parasite. Oh, my God.
Why are you laughing?
That's a great name, the castrating barnacle.
That's a serious answer.
It causes behavioral changes in the crab and arrests its reproductive development. It even tunnels its growth into the sexual sac of the crab. Talk about some parasitic manipulation. I really enjoyed the book, This Is Your Brain on Parasites, which talked about it. Highly recommended. Included are my happy face at the Maguro Museum and a Saculina manifesto. Thanks again for the awesome podcast. Anita sent some photos of herself. So she went to the Maguro Parasite Museum in Japan. She has a photo there in the front, and then a composite of various exhibits, including the crab with the castrating barnacle parasite. Mm-hmm. Daniel, can you see those pictures? I think
I'm cringing here, actually. Looking at
Beautiful specimen jars, aren't they?
Yes.
Thank you, Nita, very much. We love getting pictures not only of our listeners but also of the parasites that they see. Dickson, do you know about this castrating barnacle parasite?
I've heard about it. I'm not terribly familiar with it.
So this castrates the crab, right?
Right.
Oh boy, that would be fun to talk about. Well, not for the reasons that you're thinking, but
this week in arthropods or something, maybe.
Yeah, it's interesting. Like, why? What would be the evolutionary selection for that? Right. What would be the advantage?
Yeah,
interesting stuff.
Hard to know that. Dickson.
Brian writes, "Good day, Viro Medisite Docs. That is my own portmanteau for my favorite podcast hosts. I'm back. It's been a while since I last emailed for the case study guests, and I have been playing catch-up on the past episodes. In case you have forgotten, I am the guy that likes to listen at work and also tries to type while working. Though I think I am too old for this, as usual. I make many mistakes. The millennial generation is working, though, and I much better than in this. I'll read that again because I am of the same generation. The millennial generation is much better at this. Nonetheless, I am on my smart, trying to multitask. My guess for case 131 is paragonomyosis, caused by P. westermani. I love this parasite, and I have been interested in it since reading a case study in my undergraduate parasitology class. I think you even covered the same case from St. Louis involving P. calicati from the surrounding area, some of which I have visited during summer getaways. This parasite is acquired by consuming raw or undercooked crustaceans harboring the active metasecariae that exist in the duodenum and then burrows through the intestine, peritoneal cavity, and the diaphragm into the lungs, where it encapsulates and develops into an adult. I think it takes about 2 to 3 months to begin egg production. However, there is usually more symptoms such as abdominal pain, fever, and weight loss, and eosinophilia. So that's my guess, and even though I don't always write in, if you can call my barely coherent rambling writing, I do listen to all the Twixt series, with this being my favorite. Keep them coming, and just because it can't be said enough, be nice to Dickson. Thank you, Brian.
Dick, don't you love how our listeners have taken up your cause?
I do. Well, I'm capable of defending myself, but thank you all for your empathy and sympathy.
Defending you against my attacks.
Nah, they're blunt attacks.
Daniel.
All right. You guys did this again to see if I could pronounce swellen. Right. So apparently Sue Ellen. I'm going to ask the people when they write. Oh, look, she does right there.
She did. She gave you a pronouncer's
first an important
note.
Right. My name is pronounced Sue Ellen. Okay. If everyone can do that, that would be helpful.
Yes, that's right.
I noticed my kids have the same issue. They read so much as opposed to, I guess we're all a bit of introverts in my family. Yeah. So a lot of things we see rather than hear. And so the pronunciation I'm often entertained by, and I think they may have also inherited it from me. So, okay, Sue Ellen. I can't help. My mom decided to shove it all together and make it one word. I'm used to it being mispronounced, and I adore you guys and your show. So I'm not taking offense, just correcting.
No problem.
Loved the last episode, even though I once again failed to provide the correct diagnosis. At least I'm consistent. I'm not happy with my performance thus far, so I'm going to try to get it right this time. There is not much to go on with our Thai guy. The main symptom is that he's vomiting blood, perhaps on the location Thailand, and the patient's eating habits. I'm going with paragonomyosis or lung flukes. To quote my primary source, the Southern Nevada Health District's website, humans most commonly become infected by eating raw, undercooked, salted, or pickled freshwater crabs or crayfish that contain the parasite inside a cyst. One study found that approximately 17% of harvested crabs contain the infectious cysts. I am not going to add much to this since, if I got it wrong, I don't want to go on and on like I did last week with the wrong diagnosis. My guess could be confirmed by checking for eggs in the patient's sputum. If it does prove to be paragonomyosis, then praziquantel seems to be the drug of choice. An aside, I use praziquantel to help control large and small strongyles in my horses. Strongyles. So you got to give me pronunciation guides for everything.
Strongyles.
And you pronounce this is horses. Okay. Keep giving us these great podcasts. I listen to all of them, and I really enjoy learning all the cool stuff in them. Sue Ellen.
Nice.
So you and I, Dickson, even didn't get it right because we said Sue Ellen.
I know.
And it's Sue Ellen.
Right.
Okay. Vincent, Dickson, and Daniel, right?
Yes.
Those are easy.
Yeah.
David writes, it's our last one.
Right.
Dear hosts, after being stumped for the last few case studies, I have returned to once again partake in the parasitic puzzler. I believe the man in Thailand suffering from hemoptysis is suffering from a lung fluke in the Paragonomus genus contracted by the consumption of raw crab. P. westermani occurs primarily in Asian countries such as Thailand, China, Vietnam, and the Philippines. The CDC website notes that, quote, specialty dishes in which shellfish are consumed raw or prepared only in vinegar, brine, or wine without cooking play a key role in the transmission of paragonomyosis, end quote. And salted crab somtum seems to fit into this category nicely. Treatment for paragonomyosis includes praziquantel given at 75 milligrams per kilogram per day with 3 doses taken over 3 days. The weather has finally been looking up. It is a very pleasant 21 degrees Celsius and mostly cloudy in North Grafton, Massachusetts. Once again, thank you for the informative podcasts.
Right.
Yeah, we had good engagement there, Dickson and Daniel, don't you think?
We did.
Yeah.
We did. It was. I think we were pretty consistent, right, on the answers here.
Everybody had the right guess, absolutely.
I think so.
And it's amazing for a 4 or 5 sentence description, right?
True.
So, Daniel, did you see this man?
I did, actually, and I think the question we had is, so a lot of us, just based on the history, had an inkling of what it might be. Mm-hmm. And the next question, and we'll put this maybe to Dickson here, what would you do? Well, how would you clinch this diagnosis?
Well, I think one of the listeners wrote in and said that you should do a sputum exam to look for the eggs, but you could also do a stool exam and look for the eggs too, because most people swallow their sputum rather than expectorating it. So the eggs would be found in both places, and the eggs are very characteristic for Paragonimus. Mm-hmm. They have an operculum at one end, and they're sort of an urn-shaped.
Urn?
They look almost like an urn, a bottomless urn without a place to sit on the bottom.
I thought they looked like footballs.
No,
yeah.
those were the.
I'm an American, right?
Those were the Trichuris eggs.
Yeah, so it's
half a football.
Yeah, so it's slightly ovoid, but then someone chopped the end off of the football, right, and you've got this operculum there.
That's correct.
And so this person coughed up, and these would be sort of the subtleties. So they cough it up, and you want to, and I think Joseph brought something up here that I'm going to allude to, is so you want to not destroy these. So you want to look at them under a wet mount. So the person coughs them up, and you're seeing these eggs as we described, these operculated eggs, and they're a little over 100 microns in length. They're pretty big.
They are big.
And that could be an issue because let's say this guy comes in, right? He's coughing up blood. And in most of the world, if you're coughing up blood, you've got tuberculosis, right?
That's right.
And so what people did is they would, they take the sputum, they would do the old-fashioned, the traditional acid-fast staining, which would destroy the eggs.
Exactly.
And then let's say they didn't. Let's say use the modern acid-fast staining, which doesn't necessarily destroy them. They go to a high power, and these are so big that you don't see them.
Right.
So this is, again, one of those things where, the history not only gives you the diagnosis, but actually helps you make it because you need to know how to look at the sputum. Right. You need to know what magnification to do. You also need to know how to prepare them. So in this case, they were looked at under the proper magnification. Right. They were looked at under a normal wet mount O&P preparation. And these classic P. westermanni eggs were seen and identified.
Right. So you treat this guy, right? And now what do you recommend? Don't eat any more Sumtum?
Yeah.
And that's not gonna work.
That's not gonna work.
So, I guess as a family physician, I would suggest looking at the rest of the family because obviously everybody ate the same food. So maybe everybody has this condition and it hasn't progressed yet, or maybe the worm's located to other areas, that sort of thing. Am I not correct in that, Daniel?
No, you are correct, and that's that's one of these big issues. Is that in some of these areas, particularly the dietary preferences, there's lots of exposure. It is sort of interesting, right? Why isn't everyone in the family coming in? So there probably is a difference here in maybe it's inoculum, maybe it's a genetic predisposition, because not everyone gets this.
Right.
Once you're let me ask first, if untreated, what would happen, Daniel?
So the Paragonimus is sitting there in the lung in this cavity, and the reason he's bleeding is you're actually getting an erosion into the arterial supply in the lung, and that blood is getting in. So people can actually, if you get an erosion, you can actually bleed out. Person can really cough up a lot of blood. But as we notice, this is sort of walled off. This man is not sick. It's not a wasting disease like a tuberculosis. Nope. So I'm sure there's lots of people that actually live with this.
Yeah, absolutely.
Now, after treating him, the parasite will be eliminated, correct? Can he be reinfected?
Well, I don't think it will be eliminated. I think we get killed. Mm-hmm. So then when they die, they
calcify.
There you go. So now he's got another problem because let's say later on down the road, he does develop tuberculosis.
Yeah.
And he comes in and they take a chest x-ray and they see this big calcified lesion. What would you think of that? You might, I might diagnose it as cancer.
Yeah, I mean, I think Joseph brought up the TB part, but as you bring up, Dickson, there's this issue. Like down the road, when you see this, you know, what are you thinking? And then you've got a cavity, so could a fungus ball form in there if it really cavitates? This may scar down and not leave a cavity, which might be somewhat advantageous. But yeah, it's you know, and I guess I want to refer to this issue of, I think Joseph brought up, is it a good idea to get a PPD or a quantiferon gold along with the sputum initially? And I guess we're getting slightly outside of parasites there, but as an ID doc, I'll step to the side. So, you know, in a place like Thailand, the majority of people have been infected with tuberculosis. So I'm not sure getting the PPD or the quantiferon gold adds anything. Sure. You know, he most likely has been infected. So you already go into it with the probability that it'll be positive. Yeah. An interesting issue for people who treat TB is sometimes when people actually get active TB, a positive PPD will turn negative. And there's a lot of interesting immunology ideas on why that happens.
Could he be infected again, Dickson?
Of course.
Over and over.
We don't know much about the immunity side of things with regards to Paragonymous because it's a poorly studied infection in terms of clinical disease. And it's the animal models are there, and we could work with other systems to find out, but it's not a high priority item with regards to funded researches. So the answer is I don't know, and we should know, but we don't.
You know, what they've done in a lot of these areas, and I think, you know, in addition to giving up in some places, is tried these sort of mass chemotherapy sort of under our prevention and control. But it really, in some of these areas, it's maybe somewhat unrealistic. And so it hasn't really been as effective as some of the control of other parasitic infections.
Yeah, the reason why they would use that approach rather than trying to eliminate, let's say, the intermediate host or reservoir hosts, because there are lots of reservoir hosts for these things, right? That's how they maintain themselves in the wild. There are probably 20 or 30 different species of Paragonymous-like infections that people can encounter. There's another issue here, too, and that is that freshwater fish farming for crustaceans like shrimp and also fish, of course, cyprinoid fish, is very popular right now, and it's increasing, not decreasing. And those situations build up these sort of agricultural ecosystems that encourage peridomestic animals like cats and dogs to hang around these places because they get free food, and as a result, they defecate, and then the snails build up in these ponds, and the next thing you know, you've got cycles right next to large population centers, and it's creating problems in many directions all at once.
Is there anything that now these people are not going to stop eating their sum-tum, right?
No, they're not.
What can we do?
Because it's so good.
Because you had some.
I've had salted crab sometimes, so even when you know better, it's just so good.
I had hairy crab once when I was in Shanghai, so I was thinking about this, but you know, I went ahead. And the problem is you crack these things open with your teeth, right? A lot of people use their teeth to crack these things open, and that's where the stage of the parasite is. It's not inside the crab; it's on the outside of the crab. But because you're cracking them with your teeth.
So if you
to get to the meat
cracker,
you would greatly decrease your chances of becoming. Of course,
Daniel, how did you do it? Did you use your teeth?
You know, it was all part of the whole plate. So yes.
Oh, you did. I see. Dear.
But no, it is tough. You know, it's there are these risks that are pretty hard. You know, in a particular cultural context to get rid of, and it was interesting. I remember. So a conversation, people were talking about the reduvid bugs and why did these people build these thatched roofs? And you know, my comment was, why do people in the U.S. smoke? You know, there you go. There are certain, you know, it's easy to give advice and it's hard to follow it. And
you bet.
Is there any besides this area of the world? Where else do people get this? Many other places?
Actually, all throughout Asia.
All throughout Asia.
Yeah, this is an excellent question because depending on the species, right?
Yeah.
That's right.
So. You know, we focus a lot on the Paragonymous westermani, which is the Asian Paragonymyces. But there's also one of our emailers mentioned the Paragonymous calicate, which we discussed previously, which is indigenous to North America. There's also, I guess I should also say, areas in South America, Peru and Northern South America have pockets as well. But there's also a Paragonymous africanus, which is a Western African. Paragonimus. So this is pretty significantly distributed.
As a note here, the Paragonimus calicati was first described in a Bengal tiger in Cincinnati. Now, did the tiger actually catch it in, let's say, India, and then brought to the United States as a pet, or not as a pet, as a zoo animal, and then everybody sees this big tiger, and what do you feed a tiger? Whatever it wants.
Anything it wants.
That's right. Exactly right. So maybe did the tiger actually catch Paragonimus calicati after coming to the United States? It's hard to decide because there's no history on that. But that's whether it was the first description of calicati. And then later on they found it in people as well.
So Dickson, in addition to the shellfish, the crustaceans, right?
What
other animals harbor? That's it?
Yeah.
Not fish, right?
No, not fish. That's right. Those are other things like Clonorchus and Opisthorchus.
So here in the U.S., if one just ate raw crab regularly, one eventually would get infected?
You could become infected. In fact, there were several cases. I think I've mentioned this once before, that I'm a science consultant for Monsters Inside.
Yeah, you've mentioned it about 50,000 times.
No, I did not.
You have.
Stop it. And one of the cases that they had there was a camper from Oklahoma who developed paragonomiasis, and he caught it from eating raw crayfish.
Now, what was I going to ask? Oh, this was a good question, too. We're talking about, right here. So if I'm cleaning crabs, I should wash my hands before I touch my mouth, right?
Why would you be cleaning crabs?
Why would I be cleaning them?
Yeah.
Well, let's say you would just put them in boiling water,
exactly.
Right? But I have to pick them up out of the bag. No, I dump them in.
It doesn't come off that easy.
Touch them.
It doesn't come off that easy. No. You can touch them.
So this is not a risk factor, handling uncooked crab.
Handling, no, but putting them in your mouth and cracking the shells to get to the meat, that is the problem.
Well, I've never had the urge to do that. Do they infect lobsters?
Those are saltwater, and the answer is no.
These are freshwater crabs. Well, I'm never going to eat a freshwater crab. That would be a crayfish, basically, right?
Well, in some places, if you go to the Caribbean islands, there's tons of land crabs and stuff like that you could eat.
All right, good. Well, good job, everyone.
Yes, congratulations to everyone.
Congratulations.
That's right.
All right. Anything else, Daniel, before we move on?
No, I realized I didn't, the Bengal tiger brought up the fact that there's the Paragonimus also in the Great Lakes region. So the poor tiger could have caught it in that area. So
exactly. 'Cause who knows what they're feeding the animals at the zoo? Their composite diets that they make up from various parts of leftover animals from slaughtered animals and stuff like this. So who knows?
Exactly. Okay.
All right. Yeah. We have a paper for you.
We do.
Selected by Dickson.
I did.
It's published in Science Magazine at the end of March.
Right.
The title is Inhibitors of PEX-14 Disrupt Protein Import into Glycosomes and Kill Trypanosoma Parasites.
Indeed.
First author is M. Dawidowski. The last author is G. M. Popowitz. And these individuals are from Germany.
Yep.
A variety of institutions around Germany and also Switzerland, Basel, and NYU School of Medicine, Department of Microbiology, which is right here in New York City.
It's where Daniel got his MD degree.
That is.
This is author number 8, A. Rodriguez. Interesting. Only A. Rodriguez is from NYU, and we don't know what he did. But anyway, this is about finding ways to interfere with trypanosomes, which they say the drug discovery pipeline for trypanosomiasis is thin.
Very thin. Filled with toxic drugs as well.
Yeah, they say existing medications, suramin, pentamidine, melarsoprol, benznidazole, and nifurtimox have serious side effects and often fail to eliminate parasitemia. Let's ask the expert, Daniel. Would you concur with that statement?
I would definitely concur. And maybe I'll just mention a few of them. So we've talked in the past, right, about among the trypanosomes, Chagas. Mm-hmm. We're not even really sure that treatment with benznidazole or nifurtimox really actually changes outcomes, right? There was a study, and there's a current recommendation if you find an individual that is test positive, they've been infected with Chagas, and they took the highest risk people, and they said, we're going to treat these people just so we can show that we're going to prevent progression of heart disease. This was in a New England Journal article about a year or so back. And it actually was, let's just say, less than impressive. So there's concern that you put people on these medicines, and I say the benznidazole, you have to go through a whole protocol with the CDC because it's an investigational drug. It's not just easily available. And then you try to get them to tolerate it, and they get so many side effects. They're miserable. Exactly. And then, as mentioned, we're not even sure it does what we're hoping it's going to do. Some of these other drugs, Suramin, right? I mean, these are toxic. Pentamidine in the U.S. usually monitor someone because the electrolyte, potassium abnormalities, the EKG issues, arrhythmias. So we're left here with a bunch of really difficult drugs for diseases, I'll say particularly the American trypanosomiasis, that are becoming issues of global concern. Right. In large part through immigration, right? I mean, we now have over 300,000 people in the United States who have been infected with Trypanosoma cruzi, and we don't have a great treatment.
And I must say, yes, a wall would not prevent these people from coming in because they're not from Arab countries.
The wall is from Mexico.
That's true.
Okay.
They're not coming through Mexico either.
You want to make a wall in the ocean?
No, I was thinking of the immigration policies. I said immigration policies. So, this would not have been prevented that way. Right. And people from South America also translocate to other places in the world besides North America. So, Chagas disease via transfusion is a real issue.
Translocate is a nice word we use in molecular biology.
We
often.
do. Well, I tried to keep
and you've used it to talk about people moving.
I did.
How creative.
Thank you, Vincent.
Now, this paper is all about glycosomes.
Yes.
Can you tell us what a glycosome is, Dickson?
I can.
I bet you can, because you like these organelles, don't you?
Well, I love them. Well, I have some history here because obviously I spent some time at Rockefeller University with some really subcellular particle experts.
Organelle people.
Yeah, Christian de Duve, Miklos Müller, those people knew all there was to know at that time about lysosomes, mitochondria, peroxisomes, and of course, they didn't study the glycosome. But Miklos Müller, that is, studied the hydrogenosome in Trichomonas, which we've discussed earlier. So let's just talk about energy metabolism for a moment, because I think that clarifies what this really is all about. In mammalian cells, glycolysis takes place in the cytosol. There are no particles that say this is the glycolytic cyclosome.
Yeah.
There isn't one of those. So that's interesting because it's dispersed throughout the cytoplasm.
Not in mitochondria.
No.
Their energy, ATP synthesis occurs, right?
That is correct.
Electron transport chain and all that.
Right, Exactly right. So processing glucose from C6 down to C1 occurs in the cytoplasm, okay?
Okay.
Now, that's not so true in trypanosomes. In fact, it's not true at all. The enzymes that we associate with glycolysis, all of them, are now bunched up together in one single membrane-bound particle called a glycosome.
How big are these glycosomes?
Well, there are some electron micrograph pictures of them. They're approximately the same size as peroxisomes. So we have peroxisomes,
of course.
And you should know this, right? They're on the scale of lysosomes in terms of size.
Got it.
They're much smaller than mitochondria. And in fact, in this picture, in this article, I thought that they had some pictures of the glycosome, but
I don't think they do. But maybe, maybe to give people scale, right? Let's say, let's say you put a lymphocyte about 10 microns, right?
Yeah.
And then these are going to be they're the fluorescent ones that maybe give you a sense of them. Think of these as, if you blew that up to the size of a sheet of paper, like little sesame seeds dotted around. So they're small.
Exactly. And they have an isopycnic density that's quite unique, so that on a sucrose gradient, if you homogenize a cell and then ask the gradient to separate all these things out for you by increasing the gravity, right, you get layers in the sucrose gradient that corresponds to the nuclei at the bottom, and then mitochondria next, and then the lysosomes, and finally the peroxisomes on top. And in this case, you would get the glycosomes, where the peroxisomes would locate on the top of the gradient.
Got it. Now, Dickson, what is the advantage of having glycolysis happen in this kind of organelle as opposed to floating around.
That's a very good idea. Well, I think when you've got a small amount of cytoplasm to begin with,
I see.
If you clump your metabolic pathways together into units like these asomes, then you save room for other things because the trypanosome in a single cell has to do everything that we do in our entire body.
Okay.
Maybe one way to put it.
Now, this paper is about two proteins on the outer surface of the glycosome.
This is right.
They're called PEX proteins. That stands for, what does it stand for?
It stands for some proteins.
Peroxins.
Thank you. It's right there in front of my face.
It is, peroxins.
And there are two of them, PEX-14 and PEX-5.
Well, there are many more than that, but there are two.
In this paper.
That's right.
PEX-14 is in the membrane, and then PEX-5 binds to 14. It interacts in a protein-protein manner. And that complex then is essential for import of material into the glycosome, right?
Yes.
Enzymes from the cytosol to the organelle.
And glucose.
Cargo translocation.
It's like looking at the
translocation.
You know what?
Again, that word.
It's like looking at the International Space Station, and this arm goes out and reaches up and grabs and tethers onto something coming into it. So cargo is exactly a perfect analogy.
So PEX-5 recognizes a signal on cargo protein.
It does. It's a WXXXF.
Don't curse at me. Did you say WTF?
No.
You said WXXF. Okay.
That's right. I did. That's right.
All right. And now, the interesting thing here is that previous studies have shown that if you knock down the amount of PEX-14 RNA with RNAi,
right,
under these conditions, glucose becomes toxic.
Exactly.
You disrupt import into the glycosome, as you would expect, because you're taking away this essential component.
Yes.
And glucose becomes toxic.
Exactly.
So they say this means that this is a potential drug target.
The trypanosome develops diabetes.
Now, do you know why this is, Dickson?
I don't know why it is. No, I don't.
Why that glucose becomes toxic?
Well, glucose is an alcohol, as we know, so perhaps it has no way of disarming that characteristic of the molecule.
Well, it's no longer being metabolized, right?
That's right. It just builds up in the cytoplasm.
Or, though, actually, I think they comment on that later. Maybe there's some intermediates that get metabolized, but then it doesn't go beyond that, and they're toxic. That might be also.
Okay.
All right, so that's the idea in this paper. Can we find some drugs that interrupt the interaction between PEX-14 and PEX-5?
Right, and it's very sophisticated the way they look.
Extremely. And, it's not easy to find small molecules that inhibit protein-protein interactions.
Right.
People have tried to do this in many other systems.
Mm-hmm.
And because proteins tend to interact with each other along broad faces, and it's hard to disrupt them. But as you see here, they're successful in getting some candidates.
Indeed.
Now, the first step is to determine the structure of the part of PEX-14 that would be interacting with PEX-5, right?
Yeah.
And they do that by nuclear magnetic resonance.
Not just nuclear. They use something called solution nuclear.
It wouldn't be nuclear.
Nuclear. It depends on where you're trained, of course.
Nuclear. Solution nuclear magnetic resonance. They determine it. And they say, okay, this thing looks like the human PEX-14. By the way, humans have a PEX-14.
We do.
But they don't have glycosomes, right? Because you said it just happens in the cytosol.
They have a different amino acid sequence in the pocket that they're trying to target the drug to.
Your front or back pocket?
I think it's the front pocket.
But humans do have a PEX-14.
They do, but the sequence is different.
What does it do?
That's a good question.
Should I jump in on that?
Yes,
go ahead. Anytime. You don't have to ask.
I try to be nice. I try to just jump in periodically. So it's and this was a concern I had when I started to read this, is that there's a lot of disorders, and actually some of the stuff that my research touches on a little bit, where humans, the PEX-14 can be involved. So when we talk about these glycolytical processes, there's not only the glycolytical process, but there's also the breakdown of lipids.
Right.
So there are disorders in humans that we see. One of them sort of I'll say the one that catches my eye, the adrenoleukodystrophy, where you fail to break down lipids and they accumulate. And as these accumulate, people can develop all these disorders.
Sure. Now, that's a peroxisome function. Is that correct?
So that's exactly where we're running it. So we're running into peroxisome biogenesis disorders, and that's how these all group together. So there's a concern as you go into this that if you get any kind of crossover, are you going to start basically inducing these different issues?
Right.
So
they noticed that the two proteins, this part, they determine the structure of just the N-terminal domain. It looks similar to the human protein, but there's an additional C-terminal alpha helix in the T. brucei, PEX14. They can see hydrophobic pockets, which have 2 phenylalanine residues at the binding surface. And there are differences in the sequence, as Dickson alluded to before, with human in this area, which they say this may allow us to make inhibitors that selectively target PEX14 and won't cross-react with PEX14 of people.
Exactly.
So, I mean, it did turn out that that's true.
How lucky can we get?
Yeah, but I would say that even though it looked good, it doesn't mean you could do it.
That's right.
But it gave them some... I wonder if they made it a go, no-go decision at this point. Said,
I wonder, having done research, did they go back and do this figure?
Yeah, right.
Because sometimes you start with, finding an interesting result and, almost at figure 2, like, can we, and then saying, hey, by the way, in retrospect, this was reasonable to think we were going to succeed.
All right, so the next step is now they go do some computer modeling. They make a 3-dimensional model that mimics the binding of these aromatic residues of PEX5 WXXXF, remember, Dickson?
I do.
To the PEX14 pockets.
Sure.
All right, so they make a model of this, and then they perform in silico screening.
Yeah, that's great.
So they basically have the computer make compounds that will fit into this thing and just do it by itself. You just turn it on, you go home, and the next day you come in and it's done. You don't have to do any actual drug research yet. You're just letting the computer do it. So this
virtual chemistry.
It's great. In silico screening. And then you get a bunch of hits and you dock the best ones in. And they say visual compound inspection. That's basically, we looked at it, made sure it would,
I like that.
would fit in there.
Right.
And then the best ones these are called computationally identified hits. They were tested by labeling and doing binding assays. They have N15 labeled PEX14, and this is done by heteronuclear correlation NMR spectroscopy, which Dickson has done many times in his career.
Yeah, I have to leave early because I have to get back and make sure the machine is still working.
Right, so they're basically doing a binding assay using labeled components and using an NMR to do this.
That's right.
And so they basically have derivative number 1, which is they call it a drug-like compound. It's not a drug yet, of course, until you've got some clinical results.
Right.
But this is a compound that has moderate affinity for PEX14, and it can also disrupt the interaction between PEX14 and PEX5.
Yep.
Unfortunately, I'm sorry, so this had weak affinity, but it was toxic to bloodstream T. brucei. I was going to say, unfortunately, it was toxic to T. brucei.
They said it was for another reason. It wasn't for the reason that they designed the drug for, unfortunately.
And it was low toxicity for murine fibroblasts in human cells.
Which is their good controls. That's right.
And then they started to optimize it to make higher PEX14 affinity. They were fooling around with the double aromatic ring systems in there and adding. So they basically made a screen using 1,500 fragments to identify motifs that bind PEX14. So these are influenced by the structure of PEX5. And they say, what if we add we decorate this compound 1 and with different compounds and see if it binds better.
Right.
And they're always focusing on these hydrophobic interactions there as well. So they made derivatives 2 and 3 with an extended hydrophobic interface, therefore giving improved binding affinity and significant improvement of in vitro trypanosomiasis activity.
If anybody out there wonders why you should study chemistry, this is the best paper to read as a result
Chemistry
of
is
that.
the key.
You have to know chemistry.
No matter what kind of biology you're doing,
That's
chemistry is key. Take a lot of biology in college. And for those of you who are out of college,
go back.
review it. Chemistry is just great.
It's fabulous. I was so taken by just being able to read this as like a newspaper. You start at compound number 1 with an inhibition constant of 61.6 micromolar, and you advance to 8.43 micromolar at 2 and 3. 12 is a little bit less than 2. Then you go to 4. You're going to say something about that, I presume. The inhibition constant goes way down, and then finally here it's 0.207 micromolar as an inhibition constant. That is fabulous chemistry, and that's just by design. This is absolutely purposeful all the way through.
Yeah, they're looking at. Well, they're lucky to have the structure. It's a relatively small area.
That's right.
They're looking at
totally agree. Totally agree.
It's a hydrophobic pocket with hydrophobic amino acids going into it, so they can really mess with it.
That's right.
Yeah, they do get to 4 and 5. 4 and 5 have hydrophobic features that give really good selectivity and weaker binding to the human ortholog as well, which is
that's
really good.
Also true.
So then they took compound 5. They tested in cell-based assays against different trypanosomes. Yep. It has remarkable potency against T. brucei,
which is the cattle form of trypanosomiasis.
Even more potent against T. brucei rhodesiensis,
which is the human form of trypanosomiasis.
Well, what a great pair here. Don't you think? Isn't that great, Daniel?
I'm enjoying this dynamic.
Then they tested against the amastigote, which is the intracellular form of T. cruzi, is twofold greater than the reference drug benznidazole.
That's a big improvement.
And they say mammalian cell toxicity was not expected because there's no correlation between toxicity and PEX-14 affinity. Then these inhibitors show a correlation between inhibition in biophysical assays, the inhibition of the two proteins, and trypanocidal activity in cellular assays, which they say indicate that targeting the interaction is giving it its biological activity.
Right.
So what happens here when you inhibit this interaction? You're inhibiting translocation of the enzymes that are involved in glycolysis from the cytosol to the glycosome, and that leads to unregulated glucose phosphorylation, causing ATP depletion. That leads to accumulation of glucose metabolites to toxic levels, and that's what causes cell death. So there you go. The metabolites of glucose accumulate in the cytoplasm. Normally they'd be in the, what is the thing called again?
Glycosome.
Glycosome. I just can't remember that.
That's okay.
And they would be protected there. But this thing, so this is consistent with the RNAi studies that we mentioned originally.
Yeah, that's right.
Glucose becomes toxic. Here, when you add the inhibitor, which prevents the import, it's making glucose toxic as well.
One might propose that since the rest of the PEX-14 is similar to the human, that using interference RNA therapy might also affect the human.
If you could get it, if the sequence is similar, you could probably design RNAi to be specific. They did some immunofluorescence using antibodies against these glycosomal enzymes to confirm that the drug number 5 prevents compartmentalization.
So number 4, that's right.
Of glycolytic enzymes. Yeah. And these include, and I know everyone wants to know these names, phosphofructokinase.
That's right. These are familiar to
glyceraldehyde. Oh, I'm sorry. I'm so sorry. I have to cut that out. Glyceraldehyde 3-phosphate dehydrogenase. Hexokinase. I like that one. And aldolase. They're in the cytosol. They're not supposed to be in the cytosol.
No, they're not. They're in our cytosol, but they're not in the trypanosomes.
I thought this was interesting. I don't know if this is a small point, but as you're reading this, you start thinking that disrupting this interaction is going to prevent products from getting in to be broken down. But what we're seeing here is it's a problem with the subcellular localization of the glycosomal enzymes. The enzymes can't get into this structure to do their job.
So they just act outside and create these intermediates.
That's interesting because obviously in mammalian cells, making these intermediates in the cytosol is not an issue.
No.
Right?
That's right.
But here it is, which is curious. curiouser and curiouser.
Indeed.
Do you know what that's from?
I do.
Alice.
That's right.
Go ask her.
You've been. I've been there and done that.
All right.
The restaurant or the... I beg your pardon.
Is that your dog?
Yeah, can you hear my dog?
We
This
can.
is like NPR, right? It's got like the background.
It's okay.
That's right.
I don't mind.
No, I need cattle if we're going to talk about.
No, we should probably talk about toxicaricanus or something like that.
Throw that in.
They also find that T. cruzi amastigotes are also susceptible to these inhibitors, which we've mentioned. Right? Why is that important, Dickson?
Well, because that's the dominant form of T. cruzi's division stage.
I don't know if they know if this drug will get into a mammalian cell, right?
It would have to.
We're about to get to our
it has to
animal model. We're almost to the animal model.
No, that's right.
In cell culture, that's how you grow T. cruzi.
Yeah, so this, yes, of course. So this interface is hydrophobic, all right? And therefore, they say, this drug is hydrophobic also, so it may have unfavorable pharmacokinetics, right? So that's a problem with drug development.
Sure.
So they did some experiments to look at this, and they find that the inhibitors have good plasma stability, microsomal stability, and high plasma protein binding. So they're just kind of surprised to see that. In a murine model for infection, right?
Right.
They give inhibitor 5 did not significantly affect parasitemia. After all this work.
Right.
So they made number 6.
There you go.
with significantly reduced plasma protein binding. All right, I misspoke just now. The high plasma protein binding of number 5 is not good.
No, because
obviously
then
it's going to sequester it, right?
Of course,
and that's why it doesn't have. That's why it doesn't have good
exactly
effect on parasitemia, because we know already that it does kill trypanosomes.
But it would be effective against the African trypanosomes because they don't go into cells; they stay in the blood.
Yeah, but even in the blood, if the blood is binding the drug,
this is true. This is true.
Anyway, they made number 6, which has less plasma protein binding, and oral application 100 milligrams per kilogram twice a day for 5 days reduced or stalled the parasitemia levels.
Right.
And they only went up 4 times. What is it? Average parasitemia rose 417 and 4 times for untreated, derivative 6, and suramin treated. So suramin is better than their derivative 6. But of course, suramin is very toxic, right, Daniel?
Very.
Exactly. That's the problem. I mean, this is interesting, and we get to this very end, this table where they're trying to optimize these things, and it really is this balance between your inhibitory concentration relative to the parasites, and then the mammalian cell toxicity levels.
Yep,
that's right.
And you're running into issues because I think, as they point out, this pocket that you're going after is largely hydrophobic, right? So if you're going to get a certain drug in there, you're going to have challenges actually, getting that drug even absorbed. So you want to balance between getting in there, doing what you want it to do, but actually being absorbed, being properly distributed, and not killing the mammalian cells.
Right.
That's it.
So they say no adverse effects on the mice were observed. Of course, they could be feeling badly, but
exactly right. Exactly.
All right. So here's the summary. Now, on TWiP, people have asked for us to give an executive summary at the end. This is why this is important. All right. So this is the executive summary. What they have here is they're looking for a drug that blocks the interaction of 2 proteins that are important for the metabolism of trypanosomes. And they got an initial drug, number 1, just by using a computer to model drugs and design drugs that would block this interaction. They improved it, and they got to the point after 6 tries, probably many more, but number 6 was very effective at killing trypanosomes, blocking this interaction. And it was effective in a mouse model of trypanosomiasis.
Yep.
Correct?
Correct.
And so they call this a proof of concept study, showing that trypanosome protein-protein interactions are attractive drug targets and open the way for further development of inhibitors.
There you go.
I think it's a great idea to do an executive summary in all of our podcasts.
Here.
And that way, people could go right to it and skip all the discussion.
Or we could just read the abstract when we start.
No, not skipping the discussion, but I agree. I think that's an excellent idea to have the executive summary.
I mean, it was something this involved. It may not be familiar with a lot of people in terms of drug discovery, so it's a great idea to simplify.
Well, a few people have said, I have no idea what you're talking about. I keep listening, though, and I would love you to just explain in a few sentences. So we're going to do that on all our podcasts.
That's a great idea. That's
as long as I can remember, Dickson. All right.
That's
all right. So speaking of clinical value of drugs.
Yes.
Daniel, do you have another case study for us?
I'm all out.
Hey, one day he's going to say, I'm sorry, I have nothing.
Yeah.
And we'll wrap it up, right?
No, now let me verify. Now, the next time we record, Dickson will not be here, right? He's going to be in Africa or something. Dickson, is this true?
Yeah, this is true.
Are you serious, Dickson?
Be in South Africa.
Today's the 25th.
Sorry,
the next recording is the 11th of May.
Won't be here.
Can we come and Jan and I fly down and we can record there?
Oh, that would be great. In fact, we could find lots of interesting cases down there, too, I'm sure.
South Africa.
Yep.
What city are you going to be in?
We're going to start in Cape Town and then work our way up to Pietermaritzburg.
Is this a vacation?
It's both a vacation and a business trip because I'll be giving lectures at the KwaZulu-Natal.
I don't eat any raw crustaceans, okay?
None.
All
right. So Dickson will not be here to help us
it'll
solve.
Be you and I, Daniel. Yeah.
So our listeners, they have to do the heavy lifting, so we get this right.
I could listen and Skype in from South Africa if you'd like.
Dickson, you could actually send an email guest in, right?
I could do that.
You won't be there.
You know what?
That would be entertaining. I'm encouraging you to do that. All right. Here is our case. This is a case of a young woman. And she has young children. She has a son who's about 10 years old, and she's coming to me with a chief complaint of discomfort and itchiness in the anal area.
Discomfort.
Okay. It's itchy, basically. She reports that she has tried to look at the area, but does not see anything abnormal. She reports no complaints. She's married, no complaints from the husband or other children, so it's just her. But she does say that she's noticed the son has been scratching himself a little more than usual, and she feels like just something is not right down there. Now, another thing,
in the same area, anal area, right?
In the anal area, and he's been scratching himself in the anal area, and she reports that she feels irritated down here. Now, this is the part. Now, a lot of I'll say patients come in and they've got an idea in their head. So she says, everything was just fine. About 3 months before this happened, my sister came to visit and she has 3 young children. It's about 3 months before. And I think I may have caught something from them. Mm-hmm. Right? We don't know if this is true, but that's the story she's given us. This is a woman with no past medical history, no surgical history, is not allergic to anything. Family history, there's a father with heart disease, a mother with osteoporosis. Not sure those are related to this. This individual is not on any medications. They work out of the home. No toxic habits. There's been no recent travel. They have a dog. And they do lots of volunteering, working with children at the local church and things like that.
What country are we in? Does it matter?
We are in New York.
This
is a local case. We are in New York. No, that's an excellent question, right?
Daniel, when these 3 young children came to visit, I presume they stayed in the home?
They stayed in the home. So the sister came and visited with 3 young children.
Got it.
So like 8, 5, 3, something like that. I don't remember the exact ages, but
all right.
Preschool children.
Mm-hmm. Preschool children.
Preschool, and she has one son who is 10 years old, right?
Yes.
And he's not complaining, although she sees that he's itching himself, right? Scratching himself, right?
Yes.
Okay.
They're both itching, though.
Yeah, they're both itching.
Itching. That's right.
Itching there more often maybe than usual.
Yeah, got it. All right. Anything else you're going to tell us?
You guys have any questions? Dickson, since you won't be here, here's your chance.
Have they noticed anything unusual if they examine their bowel movements, for instance? Have they seen small whitish objects on them or something of that sort? Which is, you're going to laugh at that, but that's a common observation with the direction that I think we're heading.
Oh, okay. Interesting. So we don't have that information, or that information is not being shared.
All right. Right. Or the question wasn't asked.
And they've not eaten anything unusual lately.
No, just normal diet. No.
Is there
were there any particular things in the diet you wanted to ask about?
Raw fish,
spicy foods or stuff like this.
So there is actually a history of sushi consumption. The mother says that she does not like the raw fish, but the son likes sushi.
That's right.
These are humans. These are humans, right?
These are humans.
So another question I have is that is it always itchy, or is there a periodicity to this?
It's worse at night and in the morning.
No, I don't mean that. Not diurnal. I mean like weeks apart, days apart.
Oh, is it period? Yeah. So I didn't get that. I just got it was worse. Seems like it got itchier. When they wanted to go to bed and in the morning.
Okay.
Right.
And it may, and again, that may be that just during the day they're busy doing stuff,
Yeah.
True.
And the key here is that she thinks this coincided about 3 months ago with the visit of these kids. So this has been going on for about 3 months?
So actually it's been going on for, do we say here how long? It's been going on for a number of weeks. But there was a gap, right? I mean, there was the sister visited.
Yeah.
She was fine. Yeah. 3 months later is when the symptoms began. So the symptoms began 3 months after the sister visit.
Did she call her sister to find out if her little kids had similar itchings?
That's an interesting question.
Good question, Doc.
I'm not going to be here next week, so
you don't get to ask.
I have to save my questions now.
Question.
And the answer was?
I'll put that in the good question.
But he's not going to tell you. You didn't. You didn't ask them, right?
Oh, I did.
I'm sure he did, but he's not going to tell us.
Got it.
That'll solve it immediately.
The other question I have is the woman who came in to visit you with her 10-year-old, is she under any unusual stresses lately?
She does report being under a lot of stress, and that's interesting.
Because of things going on at work or at home, or
a lot of family stress.
A lot of family stress. Okay, that's fine. Thank you.
Are you under a lot of stress, Dickson?
It depends on when you ask that question.
I'm asking you right now.
Right now, no.
Are you okay now because you're doing TWiP?
No, that doesn't mean that at all. Sometimes in my life I've been under lots of stress. Well,
of course, but right now you're okay.
Right now I feel good. I feel
we were nice to him, I think, particularly.
We were.
I haven't been nasty to him for a while.
No.
At least a week.
No, it's
actually, people telling me to be nice had an effect.
One of the people said, "Be nice to Dickson." It's our job to be not nice to Dickson.
Yeah, that's right. They get to pick on you for making mistakes, right?
That's right.
All right. I think this is perfect. We have enough information, don't you?
I think I have enough.
Yeah. So my questions would be: one of them I thought was a nice suggestion was, call the sister and find out, is there something going on that we need to know about? Has anyone else been exposed to these individuals with similar complaints? What testing or examination should we do?
Wait, did we ask about the husband?
What did you want to know about the husband?
Does he have any itching down there too?
Yeah.
He doesn't report any.
Yeah, she said no complaints from the husband.
Okay, fine.
Poor child.
And that's the only child that they have?
And no, they actually have 2 other older kids who have no complaints.
No complaints.
Anything else you want to know about?
What about the 10-year-old in stress? Is that 10-year-old under some stress also?
I don't know.
Is the husband living with the family?
Yes.
Okay.
And what was the other thing? Oh, so not only do I want people to give us a suggestion about what it was, what it might be, how do we test for it, but I also want people to, I feel like this is hopefully a reasonably easy case. Just say that. Hopefully it's not going to, you spelled sushi with a C there, Vincent. It's with 2 S's. But just throwing that out. Otherwise, I'm going to read that and say, like,
sushi,
I'm going to say a history of sushi consumption and get laughed at. But how to treat this. And I think that that's going to be an interesting thing I want to talk a little bit about. How do we treat this here in the United States?
Yeah, it is definitely.
So this is the only complaint. There's nothing else, right? Just itchiness in the anal area. No gastroenteritis. No.
There's no secondary bacterial infections associated with the itching.
The history of the anal itch.
Right. The 7-year anal itch.
7-year itch.
No, serious. It's a more serious topic than we're making it into because
making it not serious.
The parents of children that are in this situation and they themselves that might be in that situation are driven to madness by Daniel's intimation that now what do you do? Mm-hmm. Because now what you do is it's not straightforward.
Yeah, and I think hopefully we'll talk about it. We will talk about it.
Yeah.
Daniel and I will talk about it.
You will.
Is there anyone else we should get on the show to talk about it? Just us?
You can fly me back if you'd like.
Okay. All right. Very good. We have some email to read here.
We do.
You put your glasses on, Dickson. We have 1, 2, 3.
So I can see what you're reading.
4, 5. Yeah, we have 5 emails. First one's from Anthony. In TWiP 131, it's our last episode, there was a brief discussion of arthropod illusions and delusions, the perception/imagination of being bitten by spiders and of things crawling on or in the body. I'd not thought of it in over 30 years. But I ran across something perhaps similar. One of the many things that I did in the hope, generally futile, of generating income was to run a residential exterminating business. I answered one call in nearby North Hudson and was led into a well-kept apartment. The tenant, a non-immigrant in his early 40s, explained that the place was filled with very small flies that he was unable to get rid of. There were no flies.
Right.
Guess he didn't make any money on that exterminating job. With these papers in mind, he sends a series of papers. Mm-hmm. One is called A Case of Delusional Parasitosis Associated with Multiple Lesions at the Root of the Trigeminal Nerve, and Apotemnophilia, a neurological disorder. Apotemnophilia. Might delusional parasitosis be a form of apotemnophilia? Anyone ever heard of that? Daniel, you know what that is?
No.
Apotemnophilia, a disorder that blurs the distinction between neurology and psychiatry, is characterized by the intense and long-standing desire for amputation of a limb.
Oh, jeez.
Has a neurological basis.
Goodness.
And they studied an individual in this case here. And Anthony wrote... in another paper, self-mutilation can occur in severe cases. The wounds appear in areas accessible to the patient where they have attempted to excavate the parasites.
Yeah.
So Anthony asks, is delusional parasitosis one of such curious conditions that stand in the hinterland between neurology and psychiatry? Curiouser and curiouser indeed. With an organic basis, alleviation may result from suggestion while the affliction does not.
Right.
It's very interesting.
Mm-hmm.
How did we talk about delusional parasitosis last time?
When we had Jonathan on board, we brought up Ekbom's syndrome.
That's right.
Which is it's a tough thing, I actually have to say, because you read a lot of these papers about it, and there are a group of people who have mental illnesses and who are convinced, despite evidence to the contrary, that they are parasitized, that they are infected with parasites. But it's interesting because when you read some of those papers, then you find 11% in this paper, 22% in this series actually did have a parasitic infection. So I almost think we need a larger umbrella. So, patients who are concerned that they might have a parasitic infection, and then this Ekbom's delusional parasitosis, and it's got lots of names, is maybe a smaller part of this big iceberg.
Yeah.
Dickson.
Anson writes, hello, TWiP podcasters. I've been studying Racaniello perplexum for the last 2 years. A friend recently shared with me your podcast, and I thought I would write in. Racaniello was originally thought to be Trichinella pseudospiralis, but subsequent cases showed it to be something completely different. Several papers were published on the initial case calling it T. pseudospiralis. Dave Spratt and Ian Beveridge named it Racaniello after Peter Racak, who was the first to dissect out an intact specimen, and perplexum due to the amorphous cell supporting a gourd-shaped reservoir in the rectal region containing one or more refractal thin-rimmed globules. Dave then placed it within... Thick. I'm sorry, Brennan. You're absolutely right. I'll go over that. Dave then placed it within the muspisoid nematodes due to the morphological similarities with other strange nematodes, some which are found in Australian marsupials. The muspisoid were grouped in the Duralamia nematodes. There are two families, muspisoid, which are found in mice and bats, and the Robertolfusidae, found in humans, birds, marsupials, and reindeer. I'm sure I messed up that word too. One only muspisoid example, a COX-1 from a bat, was on GenBank. I was able to sequence COX-1 and 18S, this 18S ribosomal RNA, I guess, or protein rather, for Haycocknema, and it does not group together with the bat sample. The closest match on GeneBank was a 90% similarity, which can be a problem when you're trying to pinpoint where it fits in on the nematode tree. More sequences from the different muspisoid genera are needed to help place them with higher confidence. Right now, Haycocknema falls near the oxyurids, which is the pinworm group, and rigonematids, the millipede nematodes. Interestingly, these historically have direct or insect-related life cycles. Larval Robertolfusidae stages have been identified, in quotes, in midguts of black flies in Cameroon and in midges of Australia. When you look at the known cases from an epidemiological perspective, the bushmeat hypothesis doesn't really hold up. The first patient, a vegetarian botanist in Tasmania, claims to have strayed by eating wallaby once or twice. Two other patients claim to have never eaten native animals. Perhaps the initial diagnosis of Tissus spiratus, which, by the way, was identified first in Tasmania, helped to fuel the bushmeat hypothesis in subsequent cases. More genetic evidence from a variety of muspisoids is needed to help clear it all up. Incidentally, the Haycocknema-like nematode found in the Swiss horse is most likely Haliciphalobus gingivalis, perhaps another interesting candidate for a future podcast. Very interesting, I'll just say. We had just had the first case of Haliciphalobus here in Australia a few years ago. The case in the UK with the kidney transplants is quite terrifying. I created a PowerPoint last year for a talk at the ICTMM conference in Brisbane that features both H. perplexum and Haliciphalobus. If you are interested, I would include it, but it is over 10 megabytes. Megabytes, right. Looking forward to hearing more of your future podcasts. Cheers, Ashton.
Anson.
I'm sorry.
Anson Kohler.
He's a molecular parasitologist at the University of Melbourne.
Well, no, that's exactly
Daniel, I purposely didn't ask you to read that because I thought you would have trouble.
This was horrible.
But
murdered
Dickson, it's hard. Muspisoid. And Robertolfusidae.
Yeah, Robertolfusidae.
Muspisoid. These are interesting. Did we talk about Haycocknema at some point recently?
Yes, we did.
That's why this came up.
That's right.
There's a whole world of parasites out there, Dickson, that we don't talk much about.
Tell me about
Isn't there?
Oh, gosh, yes.
Thank you, Anson.
Absolutely.
Daniel, can you take the next one, please?
Yeah, the next one is kind of curious, right?
Yeah, it is.
No.
Dear parasite fanciers, why are there two spellings of mosquitoes in Parasitic Diseases 6th edition? And so we see that there's mosquitoes, MOSQUITOES, and then there's just OS without the E.
Right.
And it looks like usually we're doing it with the E, and every so often we're doing it without the E.
Right.
So 84 times with the E.
I would say that's the correct way to do it.
with the E. Actually, I think it is with the E. Maybe this is where we need the etymology. So it looks like the without the E was dropped a few times. I'll have to do like a search through. This is from Johannan. Johannan, actually, can you email us back? And it's the times when we don't use the E. I would like to identify those and add the E.
Right.
Well, you could just search for it.
Well, other people make the same mistake, as you can see by his
just search for MOSQUITOS.
That's right.
It'll come up, Dan.
Yeah, you're right.
You'll get
you're right.
Johan is in Sweden, Dickson.
To err is human, to forgive nearly impossible.
Yes. Let me read Deborah.
Yes, please do that.
On to something. So Deborah writes, "Hello, gentlemen." I like that. Yeah. I'm neither a student, doctor, scientist, or anyone else related to your world. I am someone who has recently been diagnosed with rheumatoid arthritis. I'm 47 and was under incredible amounts of stress when my symptoms began. Now that I've been diagnosed, I've found everyone has a treatment for me. And of course, coming to you, I've been told about Hellman therapy, taking hookworm pills. I've listened to a few podcasts, and you seem like you would be the exact people to debate the implications of autoimmune sufferers to begin introducing hookworms into polite society. What do you think of this? Have you discussed this before? And I'm not seeing the podcast. Thank you, Deb. And P.S. I found you through Pinterest.
That's interesting.
Yeah, very interesting.
So I'll make it. So this morning I'm in the ICU and I'm talking to the ICU attending, and then there are three of the interns. And I was actually telling them the story about a book that I had read recently, but I probably didn't start by mentioning. I said, so there's this story where this group takes these tapeworms and they genetically modify the tapeworms so that the tapeworm can actually produce, whatever sort of you need for your health. So it's producing things for, let's say you have hypertension. So they'll genetically engineer the tapeworm for you and it secretes the hypertension medicine. Or you've got diabetes and it secretes that. And it was sort of funny. I was telling it because I thought it was entertaining, because when I was telling my son Barnaby about that I had written this author and suggested that they might enjoy reading my nonfiction book on parasitology, he said, "Oh, that wasn't true." And the same reaction by the people in the ICU. And I think people have started to think creatively. And we've, I think, discussed this a little bit, that it's the pork hookworms, right? Dickson?
Pork?
It's the pig hookworms. What are the hookworms that they've used in the treatment of Crohn's disease?
Actually, I think they've used Trichuris.
It's Trichuris that they use?
Trichuris. And the Trichuris is Trichuris ovis, which is the sheep Trichuris, and Trichuris murus, which is the mouse version.
Exactly.
But you could actually give the human version if you give a few of them, because they're not pathological in terms of their effect on us until there are large numbers of these worms. So the problem with all of it is that the immune system eventually kicks in and knocks them out, and then your therapy is over because you can't use those worms again. So we have to pinpoint what the worm is actually doing to deflect the immune response away from us and onto them, and then work on that as a solution, maybe a soluble molecule of some sort.
I think that's where it's gone a lot, is that the thought, at least from, we'll say, the pharmaceutical or the biotech companies, is that maybe there won't be a big market of people willing to ingest helminths or parasites, but they might be willing to ingest a substance, which is whatever critical substance that is manipulating our immune system. And,
that's right.
And as this person points out, with things like rheumatoid arthritis, there may be a connection to changes in how clean our world, how parasite-free our world is.
Yeah, that's true. You're right. And food allergies certainly fall into that category as well. So, I've had a lot of discussions with people about this, and I can tell you that if you have Crohn's disease, and I know that you know this also, Daniel, because you've probably treated a lot of people with it, they get driven to madness by trying to cope with a disease that is virtually eating them up. So at some point in their life, they are more than willing to swallow a few worm pills if that will help. And it's sad to see people in that condition. One of them actually worked in my office for a while, and I was besides myself with empathy and sympathy for this woman who actually was quite an athletic woman, loved to swim competitively and things of this sort, and then was crippled by Crohn's disease. Every month or two she would not show up for work, and I knew exactly what was going on, and it was very sad.
Yeah, and I would say for our emailer writing in, there's actually a researcher at the London School of Tropical Medicine who is actively working in this area. And, it'll be interesting to see where it goes. I don't think it's prime time at the moment, but
right. I agree.
I think it's a very creative, interesting approach.
It's true. The other thing is, these helminths, even if the immune system didn't kick them out, they have a finite lifespan. So hookworms live for 3 to 5 years, and that would be it. And for that time. You're symptom-free. Let's say it works perfectly. And then after that, you're right back where you started.
It's interesting. I mean, we talk about, having this nice, healthy intestinal microbiome. And as I was mentioning, I think the other night at dinner, as I said to my daughter, Daisy, everyone's talking about this. And she said, no, Daddy, it's just you and the people you spend time with. But there's
Daisy's
this idea
insightful.
about a healthy intestinal microbiome. And here, the question is, are viruses part of that? Are parasites? Are these larger eukaryotic parasites part of a healthy microbiome? I mean, that's sort of open.
Yep.
Right.
I'm pretty sure we talked about this before, Dickson. I know TWiP-22 was a hookworm episode. It might have been on there.
That's true.
But
Hellman therapy is legit. It's cleared a lot of IRBs. It's been used. It's been studied. In fact, there's a diabetes center at Columbia University where they were thinking of using it because they think that type 1 diabetes is an autoimmune disease as well. So there's a lot of interest.
All right, last email's from Dave the Sheep Shearer.
Right.
Good day, doctors. Good doctors. Thank you for the interest in my tickology. Further to the babies coming out of the cut in half tick. This happened about 15 years ago, so pre-digital camera for me and smartphone. So no pics except the ones burned into my memory chips. Mm-hmm. So here goes. The tick was in the area clear of wool on beside the ewe's udder. Yes, we have to shear right up to the udder so that the newborn labs don't suck on the wool.
Right.
It was clearly a tick, fully engorged with tiny legs sticking out on the sides. It was still attached to the ewe. This was not a spider. Very short legs sticking straight out from the body. It was in a clear area, so when I cut through it, I could see that the babies came out of the black blood coming from the cut tick. My wife just reminded me that we took a tick from our dog here at home in southern Alberta that had live babies in it. This was about 12 years ago. Further note, I am currently down with what appears to be Lyme, likely contracted 3 years ago when the symptoms started. Shearing alpacas in the Okanagan area of BC.
She was.
This year, from one alpaca, we took 20 ticks off. 5 black-legged deer ticks, 8 what were referred to as Rocky Mountain spotted, brown tick with a white spots, and the rest I couldn't ID. Wanted to take the Ziploc that the owner put the pulled ticks in so I could study, but that got vetoed. The pics I took didn't turn out, so I'm sorry, but I don't have photo proof.
Yeah. Dave
the Shearer in rainy southern Alberta. Thank you for the wonderful podcast. If I had science teachers like this in school, I would have been hooked on science. That's a very nice thing to say.
It is nice.
P.S. The owner of the alpacas was from Switzerland and had the handiest tick remover that he brought from that country. It looks like a credit card with a slit in one corner. Huh. You just put the card against the skin, lining the slit up with the tick. You then slide the card forward until the slit engages the tick. Then you can roll the card over. This action pulls the tick, and you can examine the bottom side of the card to make sure you have all the parts. I've tried the tick key, and it doesn't work near as well. Can't remember the name, but we'll try to get the name if you're interested.
Right.
Yeah, do that. Would love to see it.
Maybe this was a soft tick rather than a hard tick. I'm not familiar with the biology of soft ticks, so perhaps that soft ticks do give birth to live young, but I'm not. It doesn't register with me that ticks give off babies.
Eggs. Jonathan didn't seem to think so.
No. So maybe I'll have to do some more inquiring. We've had an entomologist on our show. We should probably go back to him, the guy from Nebraska, and ask him. Jonathan. We should ask Jonathan what's up there. What did Jonathan say?
He said he's never heard of it.
Okay. Well, then is that what you just told me?
Yeah, I did. It's okay, Dickson.
Sorry, my mind was wandering.
No problem.
That's too bad, actually, because now we're at odds with this gentleman who is.
But I like the tick removal because I think
that's not bad.
That's really important. You can actually go to the CDC site because there's so many ideas about burning ticks and covering them.
I know. You pick them off. You hold them in with a needle. You sort of work around the mouth parts. It's difficult, though. It's difficult.
Yeah, you just hold them slightly up so that there's tension, and then they will slowly. You don't want to rip them out. You want to leave parts behind. You want to just mash them or burn them.
You're right.
So I think this whole tick card thing sounds interesting.
Yeah.
Yeah. Tell us, Dave, what it is.
I think if I were inventing something like that, I would call it ticked off.
Ticked off.
Oh, I like that. That's catchy.
Should we make that the title of our episode? Ticked off?
Ticked off.
What did he call it? The tick what?
Key.
The tick key.
Tick key.
But that was what doesn't work, apparently. The tick key is commercially available.
Nah. You need something better than that.
Ticked off at the tick key. How about that?
That's right. Ticked off at the tick key.
All right.
Right.
All right. You can find TWiP at iTunes, or actually it's now Apple Podcasts. Let me change that in the show notes so it's no longer iTunes. You can find.
I did not.
They rebranded.
When did this happen?
Last week.
I've been busy.
You can find TWiP at Apple Podcasts and microbe.tv/TWiP. Consider becoming a patron of TWiP, just like the old patron of the arts, the Medici family, remember?
Sure.
Become a patron of TWiP and all the Microbe TV family of science shows. You can contribute as little as $1 a month at patreon.com/microbetv. Or just go over to microbe.tv/contribute for other ways to help us out. We'd appreciate it. We just went to Washington for the Science March, and we used your generous contribution.
Yes, we did.
Help us get there. And I left Dickson there. He's actually not here today. I left him in Washington.
That's right.
Send us your questions, comments, and case guesses to TWiP at microbe.tv. Daniel Griffin is at Columbia University Medical Center. You can also find him at parasiteswithoutborders.com. Thank you, Daniel.
Oh, pleasure as always.
And we should announce that you've also posted the Spanish edition of Parasitic Diseases, right?
Yes, we now have the 6th edition. It maybe has those misspellings, not in the Spanish version, but now we have a Spanish version of Parasitic Diseases, 6th edition.
We do.
Mosquito in Spanish it's not the same.
Hola.
How do you say it?
I don't know. Ask the Spanish expert over there. How do we say mosquito in Spanish?
People have to go to our webpage and then download
it's El
book
Mosquito.
and take a look.
El Mosquito. El Mosquito.
Well, yes, but what's the next language you're going to do? Do you have another one planned?
Good question.
We're not sure. I think we've talked about Portuguese.
Portuguese,
right?
big one, yes.
Potentially a next one.
Then you got to do Chinese, Russian.
No, we won't do Russian.
Why? Not enough people?
Not enough medical schools in Russia.
Really?
Yeah.
How many are there?
I bet you there are more than in the Dominican Republic than there are in Russia.
That's too bad.
12 of them in Dominican.
They must have a shortage of physicians in Soviet Union.
They actually
Russia.
don't, because it's not as heavily populated in some areas as it is in the beginning part.
Very good. Dickson Despommier is at trichinella.org, thelivingriver.org, also parasiteswithoutborders.com.
Indeed.
Thank you, Dickson.
Hey, I had a great time.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.